Breakthrough: CRISPR gene cuts may offer a new way to chart human genome. I'm Steve Blair. Welcome to Breakthroughs. In search of new ways to sequence human genomes and read critical alterations in DNA, researchers at Johns Hopkins Medicine say they have successfully used the gene-cutting tool CRISPR to make cuts in DNA around lengthy tumor genes, which can be used to collect sequence information. In search of new ways to sequence human genomes and read critical alterations in DNA, researchers at Johns Hopkins Medicine say they have successfully used the gene-cutting tool CRISPR to make cuts in DNA around lengthy tumor genes, which can be used to collect sequence information. A report on the proof-of-principle experiments using genomes from human breast cancer cells and tissues appeared in the February 10th issue of Nature Biotechnology. The researchers say that pairing CRISPR with tools that sequence the DNA compounds of human cancer tissues is a technique that could one day enable fast, reliably cheap sequencing of patients' tumors, streamlining the selection and the use of treatments that target highly specific and personal genetic alterations. For tumor sequencing in cancer patients, you don't necessarily need to sequence the whole cancer genome, says Winston Timp, PhD. Deep sequencing of particular areas of genetic interest can be very informative. In conventional genome sequencing, scientists have to make many copies of the DNA at issue, randomly breaking the DNA into segments and feed the broken segments through a computerized machine that reads the string of chemical compounds called nucleic acids made up of 4 bases from the formed DNA and are lettered A, C, G, and T. Then scientists look for overlapping regions of the broken segments and fit them together like tiles on a roof to form long regions of DNA that make up a gene. In their example, Timp and MD PhD student Timothy Gilpatrick were able to skip the DNA copying part of the conventional sequencing by using CRISPR to make targeted cuts in DNA isolated from a sliver of tissue taken from a patient's breast cancer tumor. Then the scientists glued so-called sequencing adapters to the CRISPR snipped ends of the DNA sections. The adapters serve as a kind of handle that guide DNA to tiny holes or nanopores which read the sequence. By passing DNA through the narrow hole, a sequencer can build a readout of DNA letters based on the unique electrical currents that occur when each chemical code letter slides through the hole. Among 10 breast cancer genes the team focused on, the Johns Hopkins scientists were able to use nanopore sequencing on breast cancer cell lines and tissue samples to detect a type of DNA alteration called methylation, where chemicals called methyl groups are added to DNA around genes that affect how genes are read. The researchers found a location of decreased DNA methylation in a gene called keratin-19, KRT19, which is important in cell structure and scaffolding. Previous studies have shown that a decrease in DNA methylation in KRT19 is associated with tumor spread. In the breast cancer cell lines they studied, the Johns Hopkins team was able to generate an average of 400 reads per base pair, a reading depth hundreds of times better than some conventional sequencing tools. Among their samples of human breast cancer tumor tissues taken at biopsies, the team was able to produce an average of 100 reads per region. This is certainly less than what we can do with cell lines, but we have to be more gentle with DNA from human tissue samples because it's been frozen and thawed several times, says Tim. In addition to the studies of DNA and methylation and small mutations, Timp and Gilpatrick sequenced the gene commonly associated with breast cancer, BRCA1, which spans a region on the genome more than 80,000 bases long. This gene is really long, and we were able to collect sequencing reads which went all the way through this large and complex region, said Gilpatrick. Because we can use this technique to sequence really long genes, we may be able to catch big missing blocks of DNA we wouldn't be able to find with more conventional sequencing tools, said Tim. In addition to its potential to guide treatments for patients, Tim says the combination of CRISPR technology and nanopore sequencing provides such depth that it may help scientists find new disease-like gene alterations specific to one allele, inherited from one parent and not another. Tim and Kilpatrick plan to continue refining the CRISPR and nanospore sequencing technique and testing its capabilities in other tumor types. Wow. I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.